Hello, in this video we will discuss about the mechanism of renin release and their function. So what is renin? Renin is basically the precursor of the blood regulator. The blood regulation precursor is the renin. When released to the blood pressure will be changed. So let's begin to understand the urinary system. So this is the adrenal gland and kidney. Kidney is basically the made up of the outer capsule and medulla and cortex, outer side and inside the medulla. Medulla and cortex is collectively combined, and cortex contain glomerulus and the Bowman capsule as well. While the loop of Henle are present in the into the medulla. So this is the ureter, and let's here is the bigger picture of the nephron, which that contain a Bowman capsule, and this is the glomerulus. This glomerulus supply blood, and filtration will be occur in the glomerulus. When the blood move from the afferent arteriole to the glomerulus, so the efferent arteriole will be constrict, and in this way the pressure of the blood will give the filtration, so the filtrate will move into the Bowman capsule into the nephron. So this is the proximal convoluted tubule, loop of Henle, and distal convoluted tubule, and this is the collecting duct. This is the anatomy of the nephron. So remember, this distal convoluted tubule is attached with the glomerulus, afferent and afferent arterioles. He is passing around here. So this is here is a bigger picture of the glomerulus with the Bowman capsule, and this is the blood supply toward the glomerulus is known as the afferent arteriole, while when leave the blood toward the vasa recta for reabsorption is known as the efferent arteriole. So this is the blood will be filter, and the filtrate will be moved into the proximal convoluted tubule, and this is the macula densa cell are present in the distal convoluted tubule, and this is the juxtaglomerular cell, which that is present between the macula densa and the glomerular arterioles. This is the Bowman capsule, and this is the juxtaglomerular apparatus it is known as, and this is the efferent arteriole. So remember, when the afferent arteriole will be relaxed and the efferent arteriole will be contract, so the blood will be flow toward the glomerulus for filtration more, so the filtrate more produced. So this is the mechanism. After the constriction of the afferent arteriole, the blood pressure will decrease, will lead to move the efferent arteriole in this way easily, and for reabsorption through vasa recta. So let's begin to understand the blood pressure drop when. So the sympathetic nervous innervation, which that is here is the juxtaglomerular cell, simply I am drawing, and this is the blood vessel. This is the efferent and afferent arteriole. So this contain baroreceptor. Baroreceptor give the signal toward the brain for sympathetic division. So the sympathetic nerve, the beta adrenergic nerve, 5-HT stimulation. So this stimulation, the sympathetic nervous system will stimulate to the juxtaglomerular cell. After the juxtaglomerular cell stimulation will release renin. This renin is the precursor of the blood pressure increasing through angiotensin 2 angiotensin 2. So this is the blood pressure will increase after the drop of the blood pressure. So in this way, when renin will release, the blood pressure will increase. When renin will inhibit, the blood pressure will be decrease. So this is the fluctuation will lead to regulation of the blood pressure normally. That is very important. So the baroreceptor stimulation is also the part of the mechanism of renin release. So how it is released, let's here is we will discuss. This is the glomerulus and Bowman capsule. So the stretches of the blood vessel which that will lead to detection of the blood flow. If the blood flow will decrease, so the stretching will be less, and after the detection through baroreceptor, not chemoreceptor, but chemoreceptor we will discuss later in the next diagram. So but you should need to understand this is also the chemoreceptor are present for the detection of sodium chloride, and the chemoreceptor is also present in the proximal convoluted tubule for the detection of sodium. If the too much sodium are present, so the reabsorption will be occur. This is the juxtaglomerular cell. Again I am drawing, and this is the chemoreceptor and baroreceptor are present. When the decreasing of the stretches of the blood vessel, it means the blood pressure is going to drop. So after the blood pressure going to drop, this baroreceptor will give the signal to the juxtaglomerular cell. So the juxtaglomerular cell is near the blood vessel, the arteriole, the efferent and afferent arteriole. So it can be easily detection will lead to trigger the juxtaglomerular cell for the increasing the renin into the blood circulatory system. So I hope you make sense. So the chemoreceptor are also present in the proximal convoluted tubule and the blood vessel. So remember, the blood vessel when drop the sodium will reabsorb more, while the increase the sodium will lead to not more reabsorption. So in this way you should need to understand that is the sodium balance is through chemoreceptor, and the juxtaglomerular cell will increase the renin into the blood circulatory system, which that is the precursor of blood pressure increase. So this is the blood vessel, that is the afferent arteriole or efferent, you can say. This is the afferent arteriole. So in this way the afferent arteriole will be constrict due to the renin production, will lead to increase the blood flow for the glomerular filtration, will lead to more filtration, more excretion of the sodium chloride. When more sodium chloride will be excreted, so the blood can be dropped, blood pressure can be dropped, and this blood pressure can be dropped by detection through chemoreceptor into the blood circulatory system for detection of the sodium chemoreceptors. And the sodium chloride are also present in the proximal convoluted tubule is the excess of the sodium chloride due to the increase of the filtration will lead to reabsorption through chemoreceptor, which that trigger, and in this way the afferent arteriole contain less amount of sodium, so the sodium can easily move into the afferent arteriole when the vasa recta will be occur. So this is the chemoreceptor. Let's begin to understand. This is the glomerular filtration, so the glomerulus and the Bowman capsule, afferent arteriole and efferent arteriole, and the sodium will be filtrate. So the filtration of sodium, and this is the juxtaglomerular cell and the macula densa cell. Macula densa cell is a part of the distal convoluted tubule, which that is we are drawing here. So this is the juxtaglomerular cells and the macula densa. This macula densa is very important for the triggering of the juxtaglomerular cell. So this macula densa cells, which that will trigger and stimulate. So in this way, when salt will be decreased into the blood circulatory system, so the chemoreceptor will detect. After the detection of the chemoreceptor, in the macula densa also contain a chemoreceptor. So in this way, the decreasing of the sodium chloride will lead to increase the macula densa cell for stimulation to the juxtaglomerular cell for increase the sodium reabsorption. So the sodium will reabsorb more, and this chemoreceptor are also present in the afferent arteriole and efferent arteriole for detection, and it is the efferent arteriole the sodium level will be dropped for reabsorption will be occur, and in this way the blood pressure should be increased. So the blood pressure should be increased. How? Because the constriction will lead to renin will produce from the juxtaglomerular cell for the constriction of the efferent arteriole, not afferent arteriole, because the less amount of filtration of sodium. So the efferent arteriole will be constrict for decreasing of the blood pressure for reabsorption, and in this way the afferent arteriole will be decrease the blood pressure will lead to absorption of the sodium. So this is the mechanism. So simply you can say that when sodium level will be dropped in the blood circulatory system will lead to constrict the blood vessel for the increasing of the blood pressure. So because the volume of the blood will be drop, it means the, so in this way here is the sodium chloride when increased, so the distal convoluted tubule which that is containing a macula densa cell which that will lead to inhibit for the production of renin by the stimulation of the juxtaglomerular cell. While the sodium chloride will be dropped, so the increasing and stimulation of macula densa cell for stimulation of the renin production from the juxtaglomerular cell. So this is the renin will move into the blood circulatory system. Let's understand the function of renin. And here is the liver which that is contain angiotensinogen protein. This angiotensinogen will convert through renin stimulation to angiotensinogen to angiotensin 1, not angiotensinogen but angiotensin 1, which that require the angiotensinogen converting enzyme ACE. So angiotensin 1 is not working too much efficiently because it is basically the inactive and it is the precursor of angiotensin 2. So the angiotensin converting enzyme will convert the angiotensin 2, and this angiotensin 2 will target the different cells and different receptor. For example, the angiotensin 1 receptor and angiotensin 2 receptor, which that is function differently. Let's begin to understand the angiotensin 1 receptor. So the angiotensin 1 receptor is baroreflex, which that is also the baroreflex pathway, and in this way the oxidative stress, aldosterone release, hypertrophy, the muscularity will be increased in the blood circulatory system if the continuously increase, and the hypertrophy of the muscle, and in this way a smooth muscle and proliferation of the cell. Vasoconstriction also is the part of the AT1 receptor, while the angiotensin 2 receptor is required. It stimulates the neuronal excitability, anti-remodeling, anti-proliferative spatial memory, cerebral blood flow, neuritic outgrowth, and vasodilation. So the vasodilation through A2R receptor and AT1 receptor is vasoconstriction. Bye.